The next speaker is Thomas Mrsic-Flogel, and hopefully I didn't massacre your last name. He'll be speaking on the functional organization of local connectivity in the visual cortex. So we're back in the visual cortex with a different range of techniques. And Thomas has pioneered this really interesting approach to bring together two-photon imaging of calcium signals on a large scale to look at sensory processing in visual cortex. And I think he's going to talk to us about some new data from that research.
Possibly new data. So thank you for inviting me. It's a pleasure to be in New York. I had I experienced wonderful things in my 24 hours here. We had a fire alarm at 5 o'clock in the morning. And then I saw the motorcade of Obama going to the UN, and at the same time, a motorcyclist trying to break through the barricade, the police barricade, who was then wrestled down by a bunch of police guys. So we don't get this kind of stuff in London. So thank you for inviting me. And I would also, at the beginning, like to thank these two people, Coho, PhD student in the lab who did all the patching work and the algorithms for alignment. You'll see that in a moment. And Sonia, who did all the in vivo work. And it's really the fruits of their joint kind of altruistic collaboration that are the data that I'm going to show you today, basically. And I would like to pay homage to Thorsten Wiesel here, and also as an introductory slide. So I'm very much interested, and the reason why I became interested in visual processing is because of the work of Hubel and Wiesel, of course. And I would like to know how connectivity in the cortex actually gives rise to the function of neurons and the population coding and even perhaps perception that these neurons underlie. So just a brief introduction. We know that sensory input flows from the retina after they see an image like this and goes into the visual cortex. And the reason I think why Hubel and Wiesel's work was inspirational is because they were the first to propose how the hierarchy of connections can give rise to function, progressively more complex functions. And that is actually what I'm interested in now, and a lot of the talks today touched on that, is simply how connectivity could give rise to any computations in the circuit. So just briefly, obviously, neurons in the retina respond to changes in luminance in very localized regions in visual space, and this information is relayed to the visual cortex where neurons become sensitive to edges, bars of different orientations, and further up the processing hierarchy at different stages of visual cortical hierarchy, neurons become sensitive to corners and contours, and eventually we get face and object recognition cells even further up the hierarchy. Okay, so the trick is to understand how these functions are generated throughout the circuit. And to do this, we must understand the connectivity. So we do have a general idea how the information flows. So let's say this is the primary visual cortex. Generally, input comes into layer 4, but not exclusively. It's relayed to layers 2, 3, and 5, where it's processed further before it's sent out to other structures, cortical regions, and so on. And of course, this processing is mediated by a whole array of different cell types, both excitatory and inhibitory. This is probably an underestimate, especially after Ed Leanstock. There's probably a lot more diversity than we crudely characterize as excitatory and inhibitory. And the trick is really to understand how all these cells really connect to each other, what their functional properties are, and to create some meaning about how computations are done in this circuit. So we do this in the mouse visual cortex, and I forced my student to record this movie by mounting a spy camera on the mouse's head while it's navigating through its natural environment, and which is a cage in this case, of course. And you can see the kind of what the pattern of input is, and this is very different from the gratings that we typically present animals and so on. So when you show such movies to an animal, in this case a nested one, and record calcium signals using two-photon imaging, and I'll explain the method in a moment, this is the pattern of activity we get. So this is maybe 60 neurons in layer 2 or 3 of primary visual cortex in the mouse. The color-coded blobs are the calcium signals originating in the somata of those neurons, and the movie is sped up about 5 times. So you can see what diversity of patterns occur during presentation of such movies. We almost never see the same pattern reappearing. Even if you show the same stimulus over and over again, there is ongoing activity in the cortex that we simply can't explain. And one of the problems with, if you're on the simple feed-forward models of visual processing, is that they can't explain a lot of the variance that the neurons exhibit, right? So in order to explain that, we need to not only understand how local neurons connect to each other, but also what the long-range influences are. And again, I'll touch on that later. Okay, so how do we do this? We take a mouse, open the skull, inject a calcium indicator dye in the primary visual cortex, And the dye is Oregon green Bapta-1, which gets captured by the cells, and it makes neurons fluoresce brighter when they fire action potentials. And the idea is that the higher the peak of the fluorescence change, the larger number of spikes the neuron fired. And there seems to be a pretty good correlation with the low firing rate regime, which most neurons, the excitatory cells in the visual cortex, utilize. And then when we show, for example, gratings drifting in different directions, we see that different subpopulations of neurons are active in response to these stimuli. And then we can use this information, extract the signals from individual cells, and see how they respond to the different orientations. So these are just 2 example cells. And then we can, for example, see this cell prefers this grading, 45 degrees going in these 2 directions. And then we can use this information to obtain both direction and orientation selectivity of these neurons in these polar plots. And from those, we can color code each pixel or each cell in the imaging region by the orientation that elicits the strongest response. And the nice thing about two-photon microscopy is that you can then do this at different depths beneath the cortical surface, and then kind of collapse all the visual responsive neurons to reveal the layout of orientation preference in this case. And so when we saw this for the first time, and I think I was inspired to do calcium imaging by Reid, by the early papers of Kenichi Oki in his lab. And when I first saw these maps, I kind of wondered, how does this work? In this field of view, which is about 300 microns, okay, yes, a cell body is 10 to 15 microns, but the extent of the dendritic tree is around 300 microns, and the axonal arborization can extend up to 1 millimeter on each side. So, and because the mouse visual cortex is only about 2.5 millimeters across, effectively each neuron can talk to any other neuron in the visual cortex. So this is quite a remarkable potential connectivity. The question is it specific, right? And you can imagine a neuron sitting here, such as, for example, this horizontally tuned neuron right in the middle, is the dendritic tree would be this big, and in principle, it connect to any other neuron. And this is layer 2 and 3. So we wondered basically, what are the connectivity rules between these neurons whose axonal and dendritic arborizations overlap. And we compare that, so this is data from Kenichi Oki when he was in Clay's lab, and if you compare that to the cat, obviously neighboring neurons, so if you have a neighboring rule of connectivity, it's kind of easy to maintain your orientation preference because all your neighbors are surrounded, and if the connectivity rules are neighboring, then you can see how that works. And also, in these higher mammals, there are long-range connections that link neurons with similar orientation preference, and they're thought to mediate some sort of contour integration, perhaps, or other kind of contextual effects. So we wondered then, what is the connectivity at this local scale? Is it selective, meaning do neurons with similar, say, orientation preferences or visual responses selectively connect to each other? Or is it random, which might mediate some sort of more interesting computations in the circuit, and the orientation tuning is derived by the feed-forward input that these neurons receive, right? Those are 2 extremes. Okay, so how do we do this? So we functionally characterize all the neurons in a small volume using calcium imaging. So we simply step down 7 microns at a time and present 2 types of stimuli, both gratings and these naturalistic movies. And from that, we get calcium signals for each of the neurons, and we can then obtain the orientation preference of all the responsive neurons, but also we compute the signal correlation. And that tells us how similarly responsive are the neurons to the natural movie. So we argue that if neurons are similarly responsive to the natural movie, they will have similar receptor fields, and this captures some other aspect than orientation preference. Okay, at the end of this experiment, in the imaging region, Sonia then injects fluorescent microspheres in red into the middle of the image region. And then she takes this anesthetized animal over to Coho. Coho extracts the brain, and he slices it for patch clamp recordings. And then he picks the slice that contains the red bead track. And what he does then, he patches neurons in the vicinity of this bead track and assays connectivity with electrophysiological recordings. And Coho then develops some image registration algorithms that enable him to match neurons in vivo to in vitro. And this is the hard bit, right? In vivo, we take a stack from the top. In vitro, we take a stack from the side, and in addition, many cells die. The slice sags in the middle. There's no blood vessels, so there's a lot of distortion in it. And despite that, we can recover the identity of about 93% of the cells that have been imaged in vivo, which is quite remarkable. So, for example, when we do this registration, we see, for example, in this case, in the in vitro slice, you see 2 astrocytes here in yellow and 1 neuron in green, and you see the same 2 astrocytes here and 1 neuron in green, and you can actually match all the neurons here. Okay, but before he does the registration, he just blindly patches in the region that we had imaged in vivo. So he doesn't know actually what the identity, functional preference of those cells was at the time of patching. And he simply goes for the healthy cells. So in this particular example, he has 4 electrodes, patches 4 neurons, and he assays connectivity by injecting 5 action potentials in one cell and seeing if there's a postsynaptic potential, excitatory potential in the follower cells. And he does that for each cell in turn. And in this particular example, out of these 4 cells that he patched, he found only a single connection from cell 3 to cell 2. And you can see these EPSPs that follow the action potentials in cell 3. And then he goes back to the in vivo map, finds the same cells, and identifies their orientation preference based on the vivo imaging. And for example, cell 4 preferred this horizontal grating drifting up and down. Cell 1 didn't respond to visual stimuli. Cell 3 had this 45-degree preference, and that provided input cell 2. He repeats this experiment. He patches another quadruplet of neurons and another in the same slice. And then he does the same thing in different slices from different animals. And in this way, he builds a statistical map of functional synaptic connectivity. All the neurons that he patched in this particular example are within 50 microns of each other. So now we can start relating connectivity to function. Okay, so the first thing we did, and to me, I think this is the most striking result. So there is a very strong correlation between visual responsiveness and recurrent connectivity within layer 2 and 3. And this is the following. So neurons that do respond to visual stimuli, it doesn't matter which stimuli, simply the ones that do, they connect to each other at a reasonably high rate for pyramidal cells, above 25%. For neurons that do not respond reliably or at all to visual stimuli, they connect to each other at very low rates. So the question is, what is the purpose of these neurons? And I suggest they may get input from some other regions, and therefore they may not be nicely correlated such that they will end up connected to each other through some activity-dependent rules. Okay, then we can relate connectivity to orientation preference, and here we found a very similar relationship to what is found for long-range connections in the cortices of higher mammals, whereby neurons with similar orientation preference, meaning that there's a 0 difference in orientation preference, they connect to each other about twice the rate as the neurons which have orthogonal orientation preference. But notice that this relationship is not exclusive, it's just preferential, so there's twice as many connections for the preferred cells preferring the same orientation. We find a steeper and a more striking relationship between connection, probability, and signal correlation. So if 2 neurons respond very similarly to natural movies, they will be highly correlated. And if neurons have 0 or slightly negative correlation, they rarely respond together to these natural stimuli, which suggests they don't have similar receptor fields. And for those cells, there's very little connectivity, those that don't respond similarly, whereas those neurons that do respond similarly, they connect to each other at almost 50% level. So this to us indicates that connectivity, local connectivity, where the dendrites and axons of these cells overlap, is preferential but not exclusive, whereby neurons form some sort of functional subnetworks, but these are not exclusive subnetworks. Okay, so then we took advantage of my very brief friendship with Ed Lean, and I said, do you have any mice? And he said, what mice? Inhibitory mice? Well, he said, you should speak to Hongkui Zheng. And then very kindly, Hongkui sent us some PV, Cree, TD tomato mice that we can then look at what their functional properties are of these parvalbumin positive fast spiking interneurons, and what the functional properties and what the connectivity is. And the nice thing about this, so if you inject the calcium indicator in green, and there's a few cells that are red, and in layers 2 and 3, most of them are inhibitory, about 60% of which are fast spikers. And if you do targeted patch clamp recordings in these neurons, you can see when we show natural movies, so this is about, I think, an 8-second segment of a natural movie response with overlaid traces of the membrane potential and spikes, you see pyramidal cells respond very sparsely, but these fast spiking PV cells respond very vigorously and very densely. And if we then quantify orientation selectivity, and similar data have been obtained also in Clay's lab, we find, just like Clay, that these fast spikers have very broad tuning to orientation. So they do have a preference, so this would be the median cell, where there is some bias towards an orientation, but generally the cells respond to all orientations, whereas the pyramidal cells are highly specific. And this is the case for most cells. So we wanted to know whether we can explain the response profile in vivo by the connectivity of these cells in vitro. And so we repeated the same experiments as for pyramidal cells, but now we try to patch one of the cells that is fast spiker. So this is an example. So FS is and 3 pyramidal cells. And when we assay connectivity, we find that all 3 pyramidal cells, which are highly tuned to orientations, and in all cases different orientations, provided input to this fast spiking neuron. And another example is shown here, where Coho patched these 3 pyramidal cells, PC1 to 3, and 1 fast spiker. Then he unpatched those cells, patched 3 more, and found that 5 out of 6 of those cells provided input to the fast spiking neuron. So this connectivity is highly dense, And this is quantified here, where the connections here from pyramidal cells to fast-spiking neurons are 87%. That's very different from the mean connectivity of pyramidal to pyramidal cells. And what's more interesting is that not only are these connections highly dense, I mean, don't forget this is the slice. We're likely cutting connections, so the likely connectivity between pyramidal and fast-spiking is about 100%. These connections are also very strong. So this is a log scale of postsynaptic potential amplitude, and pyramidal to fast-spiking connections are an order of magnitude stronger. So we think that that pyramidal cells in the local network provide such strong and dense input on these fast-spiking neurons that makes these fast-spiking neurons inherit the average orientation preference of the surrounding network. And that's why they're broadly tuned, because in the mouse, neighboring cells have different orientation preferences. And we can then kind of test that idea further by looking at the population activity of multiple cells, both parvalbumin inhibitory or pyramidal, putative pyramidal cells under different stimulation conditions. So this is spontaneous activity in the dark, and this is responses to different gratings. And what you can see is that when you just observe, so these are each of these rows is the activity of one cell, calcium signals in black, and these are inhibitory cells. You can see that these sometimes the pyramidal cells fire in bursts where several cells are coactive at the same time, and during that period, all the PV cells are coactive together. And you can see the same thing here. So the population dynamics of PV cells is that generally they tend to fire together at all times, whereas the pyramidal cells fire more sparsely, and only cells with similar tuning fire together. So this is, if you want, so this PV cell activity, which tends to be always correlated, is predicted by the connectivity that they receive. Okay, so you have asked me to speculate about what are the key questions in this field, and this is what would, I guess, in the industrial jargon, call a pipeline of research that I see developing in the next 5 to 10 years. And so if we want to understand how circuits, the connections, the cell types contribute to network computations and eventual perception and behavior of animals, these are the steps that I think we need to do. And I apologize already if this is slightly patronizing to some of you, but I was asked to do this. So the kind of questions that I'm interested in is to understand what the connectivity is between the different cell types, not just in one layer, but also across layers. So how specific are connections from layer 4 to layer 2 and 3, and from layer 3 to 5, just to understand what the rules of connectivity are. Do simple cells really project only to complex cells, or can complex cells also provide input to simple cells? Questions like that, testing some old classic ideas and see whether they're borne out in this circuit in the mouse visual cortex. And to do this, not only can we use our approach, but there are probably new approaches emerging that might really speed this up. And I think this is kind of watch this space. So using holography, using spatial light modulators, we might be able to map many connections in the same circuit, maybe hundreds of connections at the same time. And another thing that obviously, and this is Clay spearheading this effort, is to again relate, do dense reconstruction of the circuit and relate that to the function of the cells. And of course, this will provide a hugely rich data set for mining about any relationships of connectivity and function. And then based on this knowledge of the intrinsic properties of cells, their connectivity and their function, I think it's very important to do some proper biophysically realistic network modeling, because that will provide a framework into which we can integrate this information, and perhaps that framework may also give us predictions about what computations the network is capable of. And then to kind of continue along this pipeline, one can then interrogate different elements of the network to see how they influence both the network computation, but also we can update the network model based on, for example, if you take out one type of neuron from the circuit and see that one computation in there is therefore compromised both in vivo in the network model, then we'll have a pretty good idea that that neuron does this particular job in the circuit. And of course, if we want to then relate that to the behaving animals, not only do we need to observe the activity patterns while the animals are perceiving or behaving, but also we'll, ideally manipulate the different elements in the circuit to see whether we can, again, predict how the behavior will be affected based on which circuit elements we affect. And I alluded to this at the beginning. Everything I talked about right now is very local. We know, and we heard from several talks today, that a lot of the connections into primary sensory areas are not primary sensory inputs. They are motor-related inputs, they're modulatory inputs, they're feedback inputs, say, from different visual areas, even from the pulvinar. And so it's very clear that we also need to trace long-range connections. And I heard from Clay that that's exactly what they're trying to do for the different visual areas, and I think that's the right thing to do. And the last slide is there are two approaches by which these long-range connections can be traced. This is extremely neat work from Troy Margary's lab, based on the pseudo-rabies tracing that was developed by Ian Wickersham in Ed Calloway's lab. And what they did was they patched a single neuron in visual cortex. So this is a layer 5 cell. You can see the apical dendrite. And using the rabies method, I'm not going to go into detail, they could reveal the locations of presynaptic neurons which is, you can see them here. They tend to be within the same layer, quite a few cells also in layer 2 and 3, which is up here. But what's interesting, if you look here in the lateral geniculate nucleus, you can see about 8 cells that also provide input to that layer 5 cell. So if you want the canonical circuit where where LGN input only comes to layer 4 and a little bit to layer 6, is already looking mysterious because this layer 5 cell gets 8 inputs from the LGN. And so through this kind of virus circuit reconstruction, we'll get ideas about the statistics of connectivity across distal regions in the cortex. And another thing that we can do is also label locally different areas of the visual cortex with genetically encoded calcium indicators to monitor what the long-range inputs are doing while, I guess this is not visible, but you can see probably little blobs blinking, and these are the boutons of axons that are coming in from a distal region into the primary visual cortex. And then one can use the calcium signals of these individual boutons. So these are 3 examples here, and actually based on the visual input that we presented, we can actually extract the receptor field of those cells and so on. So we can actually map the types of input that are coming into the cortex, and ideally we want to do this during different behaviors to see whether different influences, how different inputs affect the processing of visual information as it's coming in from the sensory periphery. And that's it. And I would like to, again, thank Ko and Sonia, and these are all the collaborators, all contributors to this work, and of course the funding. Thanks for listening.